Hello everyone, so today we're going to talk about the histology of trachea. The trachea is a short flexible air tube about 2.5 centimeters in diameter and about 10 centimeters in length. It extends from the larynx to the middle of the thorax where it divides into two primary bronchi. The main function of the trachea is to act as a conduit for the air to reach the lungs, and in the process it also conditions the air. The trachea remains patent because of the arrangement of a series of cartilaginous ring which is present one top of the other, and this is very important because for it to act as a conduit for the air to reach the lungs, it needs to remain patent. Now if we take a section of the trachea under the microscope, we get a picture like this. So if we look at this photomicrograph, we can see that there are four distinct layers coming from inside outwards. The first layer is the layer of mucosa. This mucosa layer is made up of the epithelium and the lamina propria. Underneath the mucosa layer, we have the submucosa layer. And just outside the submucosa layer is the most distinctive part of the tracheal histology, that is the cartilaginous layer. This cartilage is a hyaline cartilage. It is shaped as the English letter C and it is deficient posteriorly, where it is replaced by the smooth muscle trachealis. The trachealis and the cartilage makes the third layer of the tracheal histology. Outside the tracheal layer, on the periphery of it, is the fourth layer, the Adventitia. Now let us take one layer at a time. First, let us talk about the mucosa. The mucosa is made up of epithelium and the lamina propria, like I mentioned. So here, in this photo, we can see that the epithelium is lined by a lot of cells. There are different kinds of cells in the epithelium, but the most numerous is the ciliated cells. These are the pseudostratified columnar ciliated cells, and the cilia plays a very vital role in the defense mechanism of the airways. The cilia is characterized by an unidirectional movement towards the epiglottis and the pharynx. Thereby it constitutes the mucociliary escalator system. So this cilia forms the mucociliary escalator system, which expels the harmful dust particles or any other particles that are trapped in the mucus of the airway out of the airway. Thereby it acts as a very important and vital defense mechanism of the airways. Along with the ciliary cells, there are other cells which are appearing empty in this picture. These are the goblet cells. Now if we look at this enlarged portion, we can see that amongst the ciliated cells we have got this empty looking cells. These are the goblet cells, and these goblet cells do not have any cilia on top of it. Now let us concentrate on this photomicrograph here. This is an electron micrograph, and here we can see clearly the goblet cells in a very enlarged manner. So these are the goblet cells, and if we look at the apical portion of these cells, we will not see any cilia. Between these goblet cells are the numerous ciliated cells. So these are the nuclei of the ciliated cells, and if you go to the apical portion of this pseudostratified columnar cells, we will see the presence of the cilia, which is forming the mucociliary escalator system. Apart from these two cells, there are other cells also, namely the brush cells. The brush cells are columnar cells which bear blunt microvilli. The basal surface of these cells is in synaptic contact with the afferent nerve endings. Hence, they are regarded as receptor cells. The fourth category of cells present in the epithelium are the Kulczycki cells, or the small granule cells. These are the respiratory representative of the enteroendocrine cells of the gut and the gut derivatives. So this is a testament of the fact that the entire airway system is an outpouching of the primitive gut or primitive gut tube. These cells are sparsely dispersed amongst the other cells. The final cell present in the epithelium are the basal cells. These are reservoirs of the cell population. So if there is any damage or any death of the cells, these cells will divide and replace those cells in the epithelial lining. The epithelium of the trachea has distinctly thick basement membrane with densely packed collagen fibers. The lamina propria is present underneath the epithelium, and it has loose connective tissue. It contains lymphocytes, plasma cells, mast cells, and eosinophils, along with fibroblasts. The lymphocytes present in the lamina propria are the functional and developmental derivatives of the bronchus-associated lymphatic tissue, which is a part of the mucosa-associated lymphatic tissue, or MALT. The submucosa is present underneath the lamina propria. The boundary between the lamina propria and submucosa is defined by an elastic membrane. However, it is not distinct on H&E preparations. The submucosa is relatively loose connective tissue, similar in appearance to the lamina propria. Diffuse lymphatic tissue and lymphatic nodules are characteristically extend into the layer from the lamina propria. The submucosa contains mucous glands with serous demileunes. Their ducts consist of a simple cuboidal epithelium that extend through the lamina propria to deliver their products to the epithelial surface. These glands are particularly numerous in the cartilage-free gap on the posterior portion of the trachea. The submucosal layer ends where its fibers blend with the perichondrium of the cartilage layer. So here in this photomicrograph we can see the epithelium with the goblet cells, then we have the lamina propria, and then we have got the submucosa. This submucosa will also contain blood vessels and lymphatics. So this entire length is the submucosa, and underneath that we have the cartilaginous layer as seen in this photomicrograph. The tracheal cartilage and the trachealis muscle separate the submucosa from the adventitia. There are about 16 to 20 cartilages, and they are shaped in the shape of the English letter C. The cartilage provides flexibility to the tracheal pipe and also maintains the patency of the lumen. It is, however, seen that with increasing age, the hyaline cartilage may be partially replaced by bone tissue, causing it to lose much of its flexibility. The final layer is the adventitia. It is the outer layer. It lies peripheral to the cartilaginous ring and the trachealis muscle. It binds the trachea to the adjacent structure in the neck and the mediastinum, and contains the largest blood vessels and nerves that supply the tracheal wall, as well as the layer of lymphatics that drain the wall. So this is all about the histology of trachea. The main thing to remember in this histology of trachea is that the lining epithelium, which is pseudostratified ciliated columnar epithelium, the goblet cells are present there, and the submucosal glands, which are mucous glands with serous demileunes, and they travel all the way through the lamina propria to open up into the epithelial surface. So this is all about the tracheal histology. I hope you like the video. If you have any doubts, please post them in the comment down below. And if you like the video, then a subscription to the channel will be fantastic. Thank you.